Placebo-controlled trials are essential in clinical research to differentiate actual drug effects from psychological influences. By using a placebo, researchers can ensure that the observed effects are due to the treatment itself, not participants' expectations. This method enhances the reliability of study results, providing a clear understanding of a treatment's efficacy and safety. A placebo is a crucial tool in clinical research, allowing us to separate genuine therapeutic effects from psychological influences. By using placebo controls, researchers can accurately assess a treatment's efficacy, ensuring that observed benefits are due to the treatment itself and not the patient's belief in its effectiveness. A placebo is an inert substance, often resembling the actual drug being tested, but without active ingredients. Commonly referred to as sugar pills, these dummy treatments have no direct biological effect on the condition under study. Their purpose is to help differentiate genuine therapeutic effects from psychological responses. The placebo effect is not merely psychological, it triggers real neurobiological mechanisms. Patients often improve because they believe in the treatment, leading to endorphin release and altered neurotransmitter activity. Remarkably, even when patients know they're taking a placebo, measurable improvements can occur, highlighting the profound mind-body connection in healing. The mind-body connection is pivotal in understanding placebo effects. These effects activate genuine neurobiological pathways, illustrating how psychological factors can influence physiological healing. This connection underscores the importance of patient belief and expectation in therapeutic outcomes, revealing the intricate interplay between mental states and physical health. Placebo-controlled trials are the gold standard in medical research. They eliminate bias and isolate true treatment effects through rigorous methodology. By comparing active treatments with identical-looking placebos, these trials ensure that any observed effects are due to the treatment itself, not psychological factors. Placebo-controlled trials employ random assignment, where participants receive either the active treatment or a placebo, ensuring unbiased results. The double-blind design conceals treatment allocation from both patients and researchers, further preventing bias. This methodology isolates true drug effects by controlling for placebo responses and patient variability, crucial for identifying genuine therapeutic benefits. Randomization and blinding are crucial in clinical trials. They prevent bias by eliminating the influence of expectations and researcher preferences. These methods balance patient characteristics like age and disease severity across groups, ensuring diversity. This increases the validity of results, making them reliable and reproducible, trusted by the medical community and regulatory agencies. Placebo-controlled, double-blind randomized controlled trials are the gold standard in proving drug efficacy. They underpin Food and Drug Administration approvals and global medical guidelines, ensuring treatments are both effective and safe. These trials eliminate bias and provide robust, reliable data essential for advancing medical science. Clinical history highlights the critical role of placebo-controlled trials in safeguarding patients. These trials have prevented the adoption of ineffective treatments and exposed ethical dilemmas in research design. They underscore the importance of rigorous testing to ensure patient safety and efficacy in medical interventions. Initial open-label studies on lithium and minocycline for amyotrophic lateral sclerosis showed promise, sparking optimism. However, larger placebo-controlled trials revealed no efficacy, with minocycline potentially accelerating disease progression. These trials were crucial in preventing the adoption of ineffective or harmful treatments, safeguarding vulnerable patients from unnecessary risks. A year-long trial with 482 rheumatoid arthritis patients compared a new drug against methotrexate and placebo. Results revealed joint damage in the placebo group progressed 4 times faster than those on active treatment, underscoring an ethical dilemma. Placebo participants faced irreversible harm while being denied effective therapies. In progressive diseases like rheumatoid arthritis, using placebos when effective treatments exist can be detrimental. Patients in placebo groups may suffer irreversible damage, raising ethical concerns. The balance between scientific rigor and patient welfare becomes crucial, as denying proven treatments can lead to significant harm. The use of placebos in clinical trials presents ethical challenges, especially when effective treatments are available. Balancing scientific rigor with patient welfare is crucial. The ethical dilemma intensifies in progressive diseases, where withholding proven therapies can lead to irreversible harm. Addressing these controversies requires careful consideration of both scientific and ethical perspectives. The ethical debate centers on whether it's justifiable to use placebos when effective treatments exist, especially in progressive diseases where withholding therapy could cause irreversible harm. Addressing patient misconceptions is crucial, as therapeutic misconception can lead patients to believe they will benefit, despite the possibility of receiving a placebo. Active controlled trials directly compare new drugs with standard treatments, bypassing placebo use. Historical controls utilize existing patient data for comparison, eliminating the need for a concurrent placebo group. Add-on designs ensure all patients receive standard care, with the experimental drug or placebo added, maintaining ethical standards while assessing efficacy. Institutional review boards demand rigorous justification for placebo use, balancing scientific need with patient risk. Patients must be fully informed about placebo risks, treatment alternatives, and potential disease progression. Safety protocols, including early escape provisions and rescue measures, ensure patient protection from extended placebo exposure if their condition worsens. Modern neuroscience shows that placebo effects trigger real changes in brain chemistry and neural activity. These effects are not just psychological. They involve measurable alterations in neurotransmitter levels and brain regions associated with pain and emotion, highlighting the complex interplay between mind and body in therapeutic contexts. Expectation activates neural pathways, enhancing perceived improvement. Conditioning from past medical experiences shapes responses. Motivation boosts perceived benefits, while anxiety reduction through treatment rituals lowers stress. The medical setting itself enhances therapeutic expectations, contributing to the placebo effect's power. Placebo analgesia activates endogenous opioids, releasing natural painkillers, reversible by naloxone, confirming opioid involvement. Placebo antidepressants alter serotonin pathways, mirroring active drugs. Neuroimaging reveals authentic metabolic and structural brain changes during placebo responses, underscoring the physiological basis of these effects. Placebo effects are not mere illusions. They manifest as measurable brain phenomena. Advanced neuroimaging reveals that placebo responses induce authentic changes in brain activity, metabolism, and neurotransmitter function, underscoring their physiological reality. Contemporary placebo trials prioritize ethical considerations by integrating innovative designs and enhanced safety protocols. These trials aim to balance scientific rigor with patient safety, ensuring minimal risk exposure. Strategies include adaptive trial designs, which allow modifications based on interim results, and the use of informed consent processes that clearly communicate potential risks and benefits to participants. Minimizing risks in placebo use involves limiting exposure periods and incorporating brief washout phases. Statistical methods can reduce placebo group sizes, minimizing patient risk. Rigorous patient monitoring with clear early withdrawal criteria ensures safety for those experiencing disease progression, maintaining scientific validity while prioritizing patient welfare. Open-label placebo studies are reshaping medical research by eliminating deception, offering transparency. Personalized placebo use, guided by genetic markers and psychological profiles, promises tailored treatments. The field is evolving, striving to balance scientific rigor with patient welfare through innovative methodologies, ensuring ethical and effective placebo application in future medicine. Placebos are crucial for differentiating genuine therapeutic effects from mind-body influences in treatments. Ethical use demands transparency, robust safeguards, and exploring alternatives. Recognizing placebo's role is vital for developing therapies that are both safe and effective, ensuring all patients receive genuine benefits.